All right. Well, hello everyone, and welcome to ASBMB Journal Club. I am Raj Mukhopadhyay, the science writer for the American Society for Biochemistry and Molecular Biology.
I'm Jeff Hunt. I'm the Society's Public Outreach Coordinator.
So ASBMB Journal Club is an interactive feature where we showcase authors who publish with the Society's 3 journals. ASBMB publishes the Journal of Biological Chemistry, Molecular and Cellular Proteomics, and the subject of today's hangout, the Journal of Lipid Research.
So today with us we have 2 authors from a recent mini-review series in the Journal of Lipid Research. We have Bill Blainer from Columbia University. Say hello, Bill.
Hi.
And we have Marit Traber from Oregon State University.
Hello, everyone.
So today's discussion will be on vitamin E, and vitamin E has been part of a 4-part review series that JLR is running. We had a discussion last month on vitamin A. Today, as I said, we're discussing vitamin E, and there'll be forthcoming review series in JLR on vitamins D and K. All these vitamins are fat-soluble vitamins, so that's why they're grouped together.
So we're going to get started with the discussion. Bill and Maret will answer questions that we have, but if you have questions out there watching, please feel free to post them on our Google Plus page. You can tweet at us at ASBMB if you use the hashtag Journal Club, and we'll try to get the answers from these vitamin E experts. So everybody ready to go?
Sure.
Okay.
Great.
So the first question is a fundamental question. What exactly is vitamin E, and how is it different from the other 3 fat-soluble vitamins?
So I'll go ahead and give it a try. Vitamin E is not a cofactor, not a nuclear transcription factor. It's an antioxidant. It's a fat-soluble antioxidant. And if I could have my first slide, which I'm not seeing my first slide, but lipid patience is a virtue.
Yep.
Well, lipid peroxidation happens in membranes where you have polyunsaturated fats. And unlike other kinds of peroxidation events, lipid peroxidation is dangerous because it is a chain reaction. That chain reaction turns out that it creates more radicals. So here you can see there's an initiating event in the presence of oxygen makes a peroxyl radical, and that peroxyl radical attacks the polyunsaturated fat. It generates a lipid hydroperoxide, which can also reoxidize, but most importantly, it creates another carbon-centered radical. So the chain reaction keeps on going and could hypothetically tear up your entire membranes. Now in the next slide, we see what vitamin E does. It reacts with that peroxyl radical 1,000 times faster than the radical can react with other fats. In the process, we make an oxidized vitamin E, which is called a tocopheroxy radical. That radical actually can then react with vitamin C, and the whole thing protects vitamin E as well as your membranes.
All right.
Bill, do you have anything to add to that?
No, Maret's the expert on this, and I simply defer to her. When I have these questions, I email her.
Okay, so one question is, how did vitamin E get discovered?
I'll defer to Maret on that one, too.
Well, I think it's one of those sad stories where there probably wasn't enough research money, and Evans at UC Berkeley was feeding rats oxidized fat, and suddenly his rats didn't have babies anymore, and the babies were being resorbed because the mothers required vitamin E. And by giving them fresh lettuce leaves, vitamin E was discovered to be made by plants, and so hooray, we now have vitamin E protecting that fetus from getting resorbed.
That's pretty important, I'd say. So In the intro to this series, Bill, on vitamin E, you called it the enigmatic one. What makes it enigmatic?
Well, for me, and this is coming from a perspective of working with a vitamin. My own work is with vitamin A. Working with a vitamin that has very defined functions. So the role of vitamin A in vision was elucidated in the 30s and 40s, and George Wald won the Nobel Prize for basically generating that molecular understanding of one aspect of vitamin A, whereas later in the late 1980s and 90s it became clear that all trans and perhaps 9-cis-retinoic acid are important transcriptional regulators. So vitamin A, and this is true for vitamin D, and the cofactor action for vitamin K, all have very defined molecular actions. Well, vitamin E, as Moret said, is an antioxidant, and I find that to be, less specific a molecular mechanism, albeit a very, as Moret presented it, a very, very plausible one. But it also is, people have looked, I think as Moret's first answer implied, for other functions. Is vitamin E a transcriptional regulator? Does it have other actions within the body? And so for a long time, people looked for other things that vitamin E might do in the body, and it's remained a very potent lipid antioxidant in lipid environments. So maybe Moret could comment on that, but since Moret has, spent her entire career, or pretty much her entire career, looking at this, and I remember discussions with Moret many years ago about whether tocopherol has transcriptional regulatory activity.
So I think what makes the whole vitamin E story so confusing is that plants actually make 8 different kinds of vitamin E. And so they make Alphatocopherol, Gammatocopherol, the tocotrienols, and all of these are really excellent antioxidants. They're fat soluble, and so people were quite confused why, if the diet has so much Gammatocopherol, and it has almost 10 times more gamma than alpha in the United States because we eat a lot of soybean oil, why is it that when you measure what forms of vitamin E are in the plasma, you only see alpha-tocopherol? Which then brings me to my next slide, because what you see on the slides are that the various forms of vitamin E are absorbed along with other dietary fats. And those dietary fats all get packaged into a chylomicron. So all of the fat-soluble vitamins, A, D, E, and K, are put into those chylomicrons. I saw my slides for a 1/10 of a second.
They're coming back.
They're back? Okay.
They're coming back. 1 second.
I don't see them on the screen. I see the smiling people from...
there you go.
Okay, okay, there you are. And this has been known for a very long time, and I'm sorry intestine decided to spell itself in an unusual way, but basically all the forms of vitamin E are being carried into the body in chylomicrons. Then if you take a look at the next slide, what you see is the magic happens. The chylomicron remnants get to the liver, and somehow the liver is putting alpha-tocopherol into VLDL. And that somehow is in fact the tocopherol transfer protein. So this was a huge step forward in vitamin E history, in my opinion. The recognition that there was a protein in the liver that grabbed onto alpha-tocopherol, put it into the plasma. The other forms of vitamin E were excreted, and so the alpha-tocopherol is maintained. So for a long time, studies in animals, if you put them on a deficient diet, it was really hard to make them deficient. Now we know if you knock out the tocopherol transfer protein, you can make the animals deficient quite quickly. And not only could you make them deficient quite quickly, it turns out that the alpha-tocopherol transfer protein, which we thought was only expressed in the liver, is expressed in the brain. And so we've done some studies in zebrafish where we knocked out the tocopherol transfer protein and the brains didn't form in these zebrafish embryos. And that gives you an idea just about the time when the highly polyunsaturated fat DHA is being synthesized is the time where you critically need alpha-tocopherol. This protein is upregulated, and if you don't have it, the brain's oxidized, they don't form, and that's why the fetus is probably resorbed.
Actually, I have a very simple question. Where does the name tocopherol come from?
So this was one of those historical things that Evans actually named the vitamin E as tokos from the Greek, meaning to give birth,
Okay.
and the OL at the end means it's an alcohol. And ferrin, I forget what that means, but it's basically to bring forward and give birth.
Huh. Okay. Sorry to interrupt you, but I was wondering where that name came from.
So I guess sort of a fundamental question. I'm not sure if we hit on this, but what are sort of the main sources of vitamin E?
Right.
You know, where can people find that in diet and natural products?
Well, you would think because it's fat, things like hamburgers and French fries, but in fact, those aren't great sources. Spinach, not such a great source either because there's not a lot of spinach fat. So it's olive oil is a good source of alpha-tocopherol. Soybean oil, corn oil, great sources of gamma-tocopherol. But if you look at nuts and seeds, not soybeans, but sunflower seeds, almonds, those are great sources. And most people don't eat enough vitamin E, so they end up having a really crummy intake. And I think it's because of all these recycling methods that we manage to, 96% of women don't get the requirement for vitamin E, and yet we seem to be walking around just fine.
So I guess, you know, where are we going with vitamin E research? You know, what's sort of the hot spot right now?
Well, I think you've probably heard on the news that vitamin E is dangerous, that it will kill you, that they're really, you know, don't take supplements because it'll make your heart disease worse. There is no scientific basis for that. So we're actually working very hard to figure out vitamin E metabolism. And so I think my next slide is really what is vitamin E metabolism, because it's not an oxidation product, but rather the body actually takes a look at vitamin E and decides it's got too much. And the way this was discovered was people who were looking for a factor in the urine that causes sodium excretion. So the head group is not changed. At least on my screen it looks like we might be missing a bond, but that's not really the case. All of the head groups are the same. There's an initial step that's an omega hydroxylation by CYP4F2, and I've highlighted that in pink so you can see that the oxidation is, or rather the hydroxylation is happening at the tail. Then there are several rounds of beta oxidation, just like you would see with a branched chain fatty acid or a regular fatty acid. And then you end up with this molecule carboxyethyl hydroxychromanol, and it's got a carboxyl group on it. So this pathway was really discovered looking at gammatocopherol, because if you eat a lot of gammatocopherol, you end up with a lot of gamma CEHC excreted in the urine, and it actually seems to increase sodium excretion. That's a little bit controversial whether it's actually the case or not, but in principle, there is some increased sodium excretion, which is probably good since your French fries are fried in the soybean oil and you're getting all kinds of Gammatocopherol. Now, we've tried to use this as a marker of nutritional adequacy. So my next slide shows you, this was a study done in collaboration with Lenore Arab at UCLA. She had trained several hundred people who were interested in their diets to record what they eat, and they did this like 8 different times, so we knew pretty well what their dietary tocopherol intake were. And you can see there's a whole lot of people down less than 10 milligrams. That is the level that's below the RDA. The RDA is at 15 milligrams a day. And you can see that that little line begins to turn up. What we have on the y-axis is the urinary CEHC. And there was some question, is the RDA too high? And I think this data says that the line for the metabolite is pretty much flat. And we did a calculation in this paper and came up with pretty much 12 milligrams of intake is where the line starts to turn up. And what that says to the average person is the RDA probably is correct, and people are eating too little alpha-tocopherol. One solution to that is to take a multivitamin that has, it's labeled 30 IUs, and that's pretty much adequate for the 15 milligrams a day. So I think in terms of how much is too much, we see that the body upregulates getting rid of too much alpha-tocopherol. And now my next slide shows you that's the tocopherol structure. But quite surprisingly, the vitamin K structures have the same tail. And one of our concerns was that maybe all of this concern about supplementary alpha-tocopherol was actually causing an increased metabolism of vitamin K. We explored that possibility, and we don't think that's the case. There does not seem to be any interaction. And what's interesting about vitamin K is phyloquinone, that structure in the middle, is present in plants. The body actually takes the K1 and turns it into MK4. And probably the most active site for this is the brain. I was completely stunned because we were trying to do vitamin E intoxication by injecting vitamin E, and what we found is we could increase the liver concentrations huge amounts, 100-fold. So we bypassed all the regulatory mechanisms for the fat-soluble absorption. But using labeled vitamin K, what we saw is in the brain, within 7 days of this vitamin E intoxication, the vitamin K, MK4 synthesis appeared to be decreased in half. The concentrations were in half. So as a fat-soluble vitamin, I think, Bill, you'd agree that this is kind of, wow, that happened way too fast. He's nodding.
The presentation you're making on the phytotail and the isoprene units in the MK4, I guess was the first time I had thought about the structural similarities between K and E. And as I was sitting here thinking, I wondered, is, one of the potential toxicities of vitamin E is in the hemolytic anemia in infants. Does it relate? Is it mechanistically related to that phytotail and how it resembles vitamin K, or is that known?
it's absolutely not known. And I think what's been known for more than 50 years is vitamin E supplements cause increased bleeding. It's been shown in dogs, cats, chickens, people, even the Women's Health Study where they were giving women, and this was 40,000 women, 600 IU of vitamin E every other day for 10 years. One of the side effects of the vitamin E was nosebleeds. And one of the benefits of vitamin E was increased, I'm suddenly going, it decreased thrombosis, but it's not arterial thrombosis, it's venous thrombosis. And I personally think one of the reasons that vitamin E is not being found to be particularly effective in heart disease anymore is that it was not working as an antioxidant, but it was working as an anticoagulant. And most men are now told, take aspirin. Take aspirin as a prophylaxis to decrease the risk of blood clotting. And so vitamin E actually works as a pretty nice anticoagulant if you don't take a lot of vitamin K. And so one of the things my lab is really working on pretty hard, especially if my NIH grant proposal ever gets submitted and the government opens back up again, that's a little push to have you all contact your congressman to get this thing finished. Okay, so back to vitamin E and K. What we'd like to see is the last slide of mine is talking about the mechanism of how this might work. And we, like I said, we've been trying for a couple of years now to try and figure it out. And all of the obvious things, the metabolism of the tail, the P450 involved, any upregulation of the P450, upregulation of biliary excretion, none of it seems to work. What we're now thinking about is it possible? This is how vitamin K works, and I understand there's going to be another JLR review article on vitamin K. So all of you who find this really interesting, stay posted because there's going to be an excellent review article coming up. But the vitamin K cycle, you have to take reduced vitamin K and oxidize it. It's a 4-electron oxidation to the epoxide. Then there's an enzyme system that takes the epoxide and reduces it back to the KH2. Now the problem with this is that what you eat with vegetables is phyloquinone, and that has to be reduced. So it's a reduction first and then an oxidation to the epoxide. Now this whole process is inhibited by warfarin. So warfarin is the most widely prescribed drug in the world because it's a great antioxidant. It interferes with vitamin K. And probably 20 years ago, Alphatocopherol quinone was suggested to be a great interferer like warfarin. Now the only problem with that is if you take vitamin E supplements, you don't make oxidized vitamin E. What you make is oxidized. Yeah, you don't end up with oxidized. You end up with more vitamin metabolite, which is the CEHC. So we're now looking specifically at this pathway. Can we unravel? Does the CEHC interfere with the vitamin K activity, which is to make gamma carboxylated proteins? And these proteins chelate calcium, and that's how they work in the blood clotting cascade. But I think it's been entirely overlooked. There's a protein called matrix GLA protein. It sits on the artery walls, and the GLA protein has to have at least 3 of those GLA molecules, gamma-carboxyglutamic acids, that chelate calcium. And so it's a little claw chelating calcium, and it prevents calcification of the artery wall. So what we see with vitamin E excess is increased left ventricular dysfunction, and we see increased hemorrhagic stroke. Both things likely due to dysregulation of calcium on the artery wall and on the brain. So that's my big guess.
Marette, can I ask a question? Presumably these studies could be done, or studies could be done in vitro, actually looking at effects of tocopherol metabolites on the vitamin K-dependent gamma carboxylase activity. I mean, has anybody done that? Because, I mean, the vitamin K cycle is ultimately driving glutamate gamma carboxylation through that enzyme. And so I guess what is the in vitro evidence? Is there in vitro evidence that that might be happening?
So that it's a very difficult assay, but that's my aim one. So I hope you're my reviewer.
Probably disqualifies me, Marette.
Darn. But yeah, that's part of the reason it's difficult is most of those vitamin K dependent proteins are low molecular weight, and now you're putting on a CO2 group, and to do this by mass spec, you blow off the CO2 groups, and so it's been a challenge for the vitamin K field. I have a collaborator, Ellen Reddy at University of Washington, who has an in vitro system done in cells with a fluorescent marker. So we have hope that we can actually tease apart this. He has tried tocopherol quinone in his system, and the concentrations are so high that they are physiologically unimportant. And I think people are unaware that vitamin K concentrations are 10,000 times lower than E in the circulation. And that's part of the, you have to recycle the K to keep using it over and over again instead of having high concentrations.
So we actually have a question that came in on our Google Plus page, and I think, Marette, you've kind of addressed this, but maybe you could clarify on this question. It's to do with physiology. The question is, does vitamin E prevent smooth muscle proliferation in blood vessels and prevent narrowing of the lumen?
There is in vitro studies looking at alpha-tocopherol and PKC activity, and you can show in tissue culture that there are decreases in cell proliferation. Unfortunately, every time these are tried in vivo in animals, so a tocopherol transfer protein knockout mouse, for example, we can't see that the PKC activity is modulated at all. So that's why it makes it challenging to study vitamin E, because all of the nice biochemistry systems that are good for studying mechanism, you end up, vitamin E is fat soluble, so you put it in an ethanol, you get really high concentrations, and things work, but as soon as you actually try and show, did that work in vivo, not so good.
Bill, do you have any further questions for Marette?
Well, I guess go back to the old question, Marette. What is the difference? Why are there different tocopherol, tocotrienol species? Do they react differently? Like, let's just restrict that to the 4 tocopherols, which differ chemically by the positioning of their hydroxyl groups. Okay, good.
I just happen to have a visual aid.
So gamma-tocopherol, does it react differently than alpha-tocopherol? Do they have different antioxidant properties? Presumably because of the location of the hydroxyl groups, they may. But
they have measurable differences. This is the vitamin E molecule. This is the tail that holds it in the membrane. This is the hydroxyl group, and this model is of alpha-tocopherol. It has 3 methyl groups. If we take this one away, now we have gammatocopherol. And gammatocopherol now has a site that could be activated. So if you oxidize this molecule and make it a radical, that activates this site and it can become adducted to all kinds of things. So it could bind to a protein. It will accept nitric oxide or peroxynitrite, so that gets people all excited about this is a reactive nitrogen scavenger. I think, though, what happens to the body is it gets really nervous about having something that can go around binding to other stuff. And so instead of having this guy binding to things you don't want to happen, the body gets rid of it by getting rid of the tail and making the whole thing water soluble. So I think the whole idea of alpha versus gamma, they're both antioxidants. They're great antioxidants. In fact, the food, if you look at your potato chips, you'll discover that vitamin E is sprayed on the potato chips to prevent the oxidation so that you can have a natural antioxidant. But alpha-tocopherol ends up being a lot easier to brown the potato chips. Therefore, the food manufacturers have found that gammatocopherol is a really nice antioxidant in oils. Similarly, tocotrienols move. They have a tail that's unsaturated, so it's straight, and so it ends up being a better molecule as an antioxidant in vitro systems. So why the body wants it, I think, is because of the possibility of making toxic adducts.
All right, so I think we're heading towards the end here, and we're not getting any more questions. So I'll give you both sort of one last, wrap-up word about vitamin E. Sort of what's coming next. What summarizes this all up.
Like, what are some of the grand challenges in vitamin E research, for example, for the next 5 years?
I think the most exciting thing that's coming is that vitamin E looks to be critical in the brain. You see in Alzheimer's patients, they have free iron. Free iron initiates lipid peroxidation. You see in the zebrafish embryo studies that is critical for the brain. Danny Manner has done some nice work looking at the tocopherol transfer protein in the brain. So I think neurology is the wave of the future.
I would actually say something more general. Having grown up in vitamin A, and 30 years ago, the notion that vitamin A is potentially preventative of so many chronic diseases. The same holds true for vitamin E. The same holds true for vitamin D. That I guess the issue is now, for all of these fat-soluble vitamins, will there be strong studies showing this disease preventative effect when populations are broken down based on genotypes or based on their genetic background, that certain people benefit from certain micronutrients, as is the case for vitamin E in cardiovascular disease. People who have a particular genotype for a particular gene benefit, whereas the rest of the general population may not. So it seems to me that very specific gene micronutrient interaction is probably very important and a grand challenge for all of the fat-soluble vitamins in the future.
I would agree with you there, and I'd like to put in a plug for vitamin K, that if you have adequate vitamin K, then having lots of vitamin E won't hurt you.
Right. And so another plug for vitamin K will be the upcoming mini review series in the Journal of Lipid Research on vitamin K, and we will likely see Bill again doing another one of these Google Plus Hangouts.
Well, I would love to do it. Would definitely love to do it. It's a pleasure.
Great. Well, it's been a pleasure having you both. Thank you so much for participating. Thank you, everybody online who was watching and following along. We will be back in future months with another episode, so stay tuned to our website, www.asbmb.org. Visit the Journal of Lipid Research at jlr.org online and look for the mini review series. And we'll say good night and good luck. So thank you all.
Thank you.
Thank you.
Bye-bye.